guys, welcome to my channel Megha Arun, and today I'm going to start the chapter Sexual Reproduction in Flowering Plants, this topic called Microsporogenesis. It's a very easy chapter, and by the end of this lesson, by the end of this video, I'll provide you with a question to which you can give me an answer in the comment section below. Okay, so since we're learning the chapter topic Microsporogenesis, we must know, all of you might have the knowledge to understand that microsporogenesis is the production of male gametes, because microspore is male gametes and genesis is production. Awesome. So male reproductive structure is called androecium. So androecium consists of stamen, which all of you knows pretty well, and stamen consists of filament and anther. Right, so filament, all of you know that it is this stalk which goes, and anther is this bilobed structure like this. This is anther and this is filament, this is stalk-like. Okay, so the end of this filament, the proximal end of this filament, gets attached to the thalamus of the flower or the petals of the flower. It gets attached to the petal or the thalamus, the proximal end of filament. So if you want to see the diagram from the NCERT textbook, it is right over here. And this long slender stalk is the filament, and this structure is the anther. Let's talk about anther in detail. The speciality of anther, or the structural speciality of the anther, is that it is bilobed. It's actually an MCQ, bilobed and dithecous. Now, what does these two words mean, bilobed and dithecous? It's very simple. See, this is how a normal anther looks, right? This is how anther looks. See, this is one, and behind it there is another one more part like this. So both are two lobes. So two lobe here means bilobed, or in one lobe, एक लोब के अंदर हमारे पास दो थीका होती है. There are two theca inside one lobe. Understood? So let's give the lobe with pink color. As it's very important for you to understand the concept, that's why. And with orange, I'm going to specify the theca. Right? So, एक anther में दो लोब होती है. There are two lobes and four theca, right? Four theca. So it's also known as dithecous. You might wonder what theca is and why we are learning about all these stuff. You'll come to know that. So एक theca के अंदर, inside one theca, from here I'm taking one theca, we have the hero of the story. कौन? Microsporangia. The purple ones represent Microsporangia. Is that clear? Yes, it should be. Microsporangia, which further develops into pollen sac, but as of now, let us take it as Microsporangia. So, एक थीका के अंदर दो sporangia होती है, और उसी वजह से, just like that, there are four theca, right? It's dithecous condition. So I say, this is how it looks. Two lobe, four theca, and inside each theca there are two microsporangia. This microsporangia develops into pollen sac later. Right, I hope this is clear, as there are MCQs coming which shows that anther's condition is bilobed and dithecous. You have to memorize that, okay? There is no need of memorization because it's clear by the diagrams. Now let us study the structure of Microsporangia. Guess structure of Microsporangia. So first of all, I'm just gonna talk about this diagram over here. This one, this one I'm gonna talk about. Now there are mainly 4 layers. Okay, 4 layers are there, from which the first 3 layers, I'm gonna say first one is epidermis. Epidermis जो हमारी skin पे होती है, the same thing, epidermis होती है. Epidermis, I'm gonna give the color this one. So let this be the Microsporangia's wall. It's kind of spherical, So epidermis. We're gonna draw the second layer, which is endothecium. Epidermis के बाद endothecium. Endothecium we're gonna give orange color. So it's also a single layer which goes like this. It's just for your understanding. I don't think so it looks like this in real life. So yeah, that is endothecium. And thirdly, we have the middle layer. So middle layer is usually 3 to 4 in number. There are 3 to 4 layers, and we're gonna give it blue color. So there is like 3 to 4 layers. I'm just gonna give it like a rough figure. 3 layers: epidermis, endothecium, and middle layer. So first these 3, there are one more too. There is one more too, but the first 3 is useful in protection of the microsporangia. पहली तो ये है कि protect करती है ये. And the second one is dehiscence. Dehiscence यानी कि छड़ना. Pollen grains का छड़ना is known as dehiscence. It will fall out. Protection and dehiscence is the function of epidermis, endothecium, and middle layer. Middle layer is 3 to 4. Okay. Next, now we have the most important, very very important, tapetum. Now tapetum we're gonna give like a green color, like this. I'm gonna tapetum, and tapetum has a multinucleated condition, so you have to understand from it. Okay, it's multinucleated. So tapetum is nutritive. It provides nutrition to the microspore or the microsporangia in the middle. The sporogenous cells here are sporogenous cells too, which I'll show you. But tapetum is a nutritive cell here. Nutritive cell. It has dense cytoplasm. This is very important. Dense cytoplasm and multinucleated condition, multinucleus. Okay, so tapetum is nutritive. It has dense cytoplasm. It is multinucleated. Okay, tapetum provides nutrition to the layer, and it also secretes. It helps in secretion. Normally cells which help in secretion has dense cytoplasm and multinucleated condition. That's how you remember. It helps in secretion too. Okay, now we have homogeneous cells. Homogeneous cells means एक तरह के cells which are compactly arranged in the microsporangia, known as sporogenous tissue. So sporogenous tissue we're going to give brown color. Okay, this doesn't look homologous in nature, but it is. So this compactly arranged homogeneous tissue, or the homogeneous cells which are compactly arranged, forms the sporogenous tissue. और sporogenous tissue की cells develop into microspore mother cell. So this is also pretty important, that sporogenous tissue develops into microspore mother cell. So mother cell, we studied last year also that microspore mother cell or megaspore mother cell undergoes division to form the gametes, right? It is the cell in which meiosis happens. So microspore mother cell में meiosis होती है, meiosis to form pollen grain or microspore. Okay, तो sporogenous tissue से microspore mother cell बनती है और उससे हम pollen grain बनाती है. Okay, Now let us explain. This is basically what microsporogenesis means, how microspore is made. But let us see a little bit more about microsporogenesis. Microsporogenesis. So first what happens is that microspore mother cell, right? I'm going to write MMC. Don't confuse it for megaspore mother cell. In this case, we're just talking about stamen, so it's just microspore mother cell. And we're going to draw it. It's just, like a rough representation. Don't take it so seriously. Let this be microspore mother cell. And MMC से क्या होती है? Meiosis 1 happens. Meiosis 1. And it forms like this. Meiosis 1 is done. But see, meiosis mein at the end we should have 4 cells, so 4 daughter cells. So meiosis 2 happens. And we get a stage like this. So यहाँ से डिविजन हुआ पहले और यहाँ से डिविजन हुआ. So we have 4 daughter cells now. इस कंडीशंस को हम क्या बोलती है? माइक्रोस्पोर टेट्राड. माइक्रोस्पोर टेट्राड 4 होती है ना यहाँ पे, 4 टू बी फ्यूचर pollen grains होती है, तो there are microspore tetrad 4. Now what happens is that secretion of wall secretion होती है, dehiscence होती है, तो it gets separated into 4 pollen grains. 4 pollen grains or 4 microspore. Okay, so एक microspore mother cell से 4 microspore, we make 4 microspore from 1 microspore mother cell. That is very clear, right? So numericals may come that अगर 2 microspore mother cells है तो कितनी microspores बन सकती है? अगर 2 है, 1 से 4 बनती है, तो 2 से कितने बनती है? 8. That's a simple numericals आती है उसमें से. And now let us take each pollen grain in detail and let's study about its structure. Pollen grains. Now this is a very simple topic. It's just, the class is gonna end soon. So the pollen grain is equivalent to the male. The pollen grain is equivalent to male gametophyte. Pollen grain is spherical in structure. वो आप बचपन से ऐसे ही इमेजिन करते हैं ना, spherical इमेजिन करते हैं हम. और इसमें there are 2 layers, 2-layered wall, let's say. One is very important, so exine, and the other is intine. Actually, it's not E, it's I. Intine and exine. Exine is the outermost covering, and intine is the innermost covering. Int, I, N, right? So in, and X तो आपको पता ही है, it's out, X out, and in, inner. Intine is inner. So let's learn about the structure. Exine is made up of a very important substance, very, very important. It's called sporopollenin. Sporopollenin is the hardest organic substance which is ever known. There is nothing more harder than sporopollenin as of now, because there is no enzymes which has been able to degrade this substance called sporopollenin. So sporopollenin is the hardest organic substance which is present in exine. Okay, exine में sporopollenin होती है. And if this is हमारा pollen grain, nucleus है यहाँ पे, intine will give with a lighter color. So ये है intine, and we're gonna draw exine also now. Exine will give blue. So exine, there is like some places where exine is not present. Like exine तो होती है, sporopollenin is not present. See यहाँ पे this space ना, I haven't colored it blue because यहाँ पे sporopollenin absent होती है, यहाँ पे absent होती है, यहाँ पे absent होती है. here. So like there are few places where the sporopollen is absent, and that place, that apertures is known as germ pore. और वहाँ से pollen tube निकलती है, like pollen tube ऐसे निकलती है germ pore से. Okay, so germ pore में sporopollen absent होती है. Okay, next one. Now intine के structure देखती है. Intine is made up of cellulose and there's an ant. Okay, cellulose and pectin. This is also important. In intine के structure भी पूछते हैं वो लोग कब. It's cellulose and pectin. Exine is sporopollenin. Intine is cellulose and pectin. When pollen grains mature, there will be two kinds of cells will be formed. Those are vegetative cell and generative cell. Vegetative cell is comparatively bigger than generative cell. Generative cell is very much small and it is spindle shaped and it floats on the cytoplasm of vegetative cell. So generative cell is very much smaller than vegetative cell because it's spindle shaped and it floats on the cytoplasm of vegetative cell. I'll show you the diagram. It's given in our textbook itself. See this one, this small spindle shaped cell is generative cell, which is very small compared to this big vegetative cell, right? So vegetative cell has irregular nucleus. It's large and irregular, and it is also the nutritive part. Vegetative cell, so by the name itself you can understand that it provides nutrients to the pollen grain. It has food reserves in it. Okay, so in majority of the plants or angiosperms, let's say in 60% of angiosperms, what happens is that this pollen grain is released in a two-stage, right? Two-celled stage. There are two cells, vegetative and generative. And in leftover 40% of the plants, 3-cell stage में release होती है, 3-celled stage. So in 3-cell stage, what happens is that the generative cell, it forms two male gametes. It forms two male gametes. So in 3-cell stage, two male gamete plus one vegetative cell will go. Okay, 2-cell stage majority, there is vegetative cell and there are vegetative cell and generative cell, whereas in 40% of angiosperms 3-cell stage में release होती है, where the generative cell divides into two male gametes and one vegetative cell. There is something called a pollen allergy, which I think I might have. Pollen allergy means that you might get severe asthma or bronchitis when a pollen falls on you or you smell pollen or something like that. So first India में कैसे होती थी that wheat was imported. Wheat के साथ there was a contaminant in wheat. Contaminant थी ये. कौन सी contaminant? It is known as Parthenium. Let me write it in a darker color. Parthenium, or also known as carrot grass. Parthenium or carrot grass. The pollen grain is used in many industries. Like it is used as a food supplement. Food supplement जैसे use करती है Western दुनिया में, and athletes use it. Athletes use it, and race horses, athletes, सारे लोग use करती है, so that they get the stamina. Viability. So viability means that कितने देर तक और how many amount of time can this pollen grain be, functional. कितने समय तक ये एक ovum को fertilize कर सकती है. Okay, till how much time it has the ability to fertilize an ovum and form a zygote. That is viability. Basically, कितने समय तक ये functional होती है. That's it. So rice or wheat में, rice and wheat, it's 30 minutes. Whereas for some other families, it can even range up to months. And you can also preserve pollen grains, which is really like interesting to know. at least for me. Like, we can preserve it. It is known as cryopreservation. Okay, cryopreservation in liquid nitrogen in minus 196 degrees Celsius. And these preserved pollen grains will be stored in, like how we have sperm banks and blood banks, there are pollen banks too, which I didn't know. Pollen banks. And yeah, say if any agriculturalist or botanist wants to take some pollen grains and use it for some crop improvement program or something like that, they will be able to do it. Pollen banks. So at the end, I promised you that I'll show you one question and which you will try to answer in the comment section below. I'll show you. As it's my first class, I know there might be some mistakes or something which you might not understand, so be free to ask me in the comment section. This question you can give me the answer for. Thank you.